Welcome back, everyone. So for this week's lab, we're going to be working on protein expression and cell lysis. So what I'm going to talk about first is how cells kind of express a protein through a process known as induction. So what induction means is in the cell, there are certain genes that have to be activated by, certain environmental factors. And when these genes are activated, specific proteins will be expressed and made. So we can actually kind of utilize this in the lab, and we can control this induction process to mass produce a certain protein that we need. And for us, that's our beta-gal protein. So to do this, we're going to utilize a very special strain of E. coli cells. So these are E. coli BL21D E3 cells. So I briefly mentioned this last week, but what this cell line is, they're still E. coli cells, but they've kind of been manipulated to do something slightly different than normal E. coli cells. So we can have E. coli BL21 cells just on its own. And what the BL21 means is that these E. coli cells lack proteases, and because of this, proteins don't get degraded as easily, which is good for us if we want to mass produce a protein. And also they are very simple to use when we want to express something through the lac operon. And you'll see why that's important later on. As for the DE3 part of the E. coli BL21 cells, the DE3 kind of represents that these cells encode for a T7 RNA polymerase. And that T7 RNA polymerase is what will actually allow us to express our protein of interest. And again, as we go further on, you'll see what I mean by that. So before we can actually even express a protein, the first thing we need to do is actually grow the cells. So in order to grow our cells, what we do is we usually just inoculate them in media and then let them grow at 37 degrees Celsius for a set amount of time. And during this time, you can see that the bacterial growth will have, 4 major phases. At the beginning, we have our lag phase, where the cells are kind of still getting used to their environment. Eventually, we move on to the exponential phase, where the cells are rapidly growing and taking up all the nutrients until eventually all the nutrients are depleted. At this point, we enter the stationary phase, where the cells are kind of just hanging about. There's not really any nutrients, so nothing's driving them to grow, but that means they're kind of just sitting there until eventually they start dying off in their death phase. So in order to have a good protein expression, we actually want to hit this area, the exponential phase. So the way we can kind of determine this scientifically is by analyzing our cell growth using a spectrophotometer. So we commonly like to look at the OD, or the optical density, at 600 nanometers. And when the spectrophotometer gives us a reading of about 0.6 to 0.8, this is a good indication that we're in the exponential phase, and we can now actually induce the cells and force them to express our protein of interest. So in order to kind of express a protein of interest, it's very important that you guys understand how the lac operon works. So the lac operon is an operon found in E. coli cells. So these are naturally in the E. coli cells. These were in our E. coli BL21 cells, and we didn't add this, it's just there. And what this lac operon is supposed to do in nature is it basically breaks down lactose, so a disaccharide, into its monosaccharide, glucose and galactose, by transcribing and translating the LAC-Z gene into beta-gal. Don't get this confused with the beta-gal we're trying to make. This is just the beta-gal that is naturally made from the lac operon. So how does it do this? So basically, when the cell has lactose present in the environment, it will actually change the configuration of lactose into allolactose. So same chemicals, just a different isomer. allolactose is able to bind to a repressor that is stuck on this operator right here. So normally, if allolactose was not present, RNA polymerase might bind to the promoter, but it cannot progress further due to the repressor. When allolactose does bind to the repressor, it gets knocked off, allowing for the RNA polymerase to continue and transcribe the LAC-Z gene, which makes the beta-gal, and then this beta-gal can break down the allolactose. This is how the cells basically get their energy. This is how they get their glucose. So remember how I was talking about the DE3 side of this of these E. coli cells? So what this means is that somewhere in the lac operon, it could be at the beginnings, could be at the end, I'm not entirely sure, but somewhere in this genome is a gene for the T7 RNA polymerase. Meaning when we add lactose and knock off the repressor, not only will the RNA polymerase transcribe the LAC-Z gene, LAC-Y gene, LAC-A gene, but it will also transcribe this T7 RNA polymerase, which will eventually be made into the T7 RNA polymerase protein. So why do we need to know that? Why do we need this T7 RNA polymerase? It's actually how we're going to get the protein from our plasmid. So this is kind of how the mechanism is working. We're trying to actually utilize, or we're trying to get these T7 RNA polymerases from the gene of the E. coli cell. And in our plasmid, our promoter region is actually specific to the T7 RNA polymerase. Meaning, when the genome of the E. coli makes the T7 RNA polymerase, the T7 RNA polymerase can then come to our plasmid, bind to the plasmid, and transcribe our gene of interest. So here it's the GFP or BFP protein, but for us, that is our beta-gal gene. So one more time, we add lactose to basically knock off the repressor and activate the lac operon. The RNA polymerase is able to bind to the lac operon, transcribe the entire operon, including our T7 RNA polymerase gene. The gene is then made into a protein that can bind to our plasmid, and from the plasmid, the T7 RNA polymerase can transcribe our gene of interest, which is beta-gal. So this is how we're getting our beta-gal. Yes, the lac operon has its own beta-gal, but this will make significantly more. So this is how we're going to get the majority of that beta-gal. So something I don't know if you guys saw on that last slide was a compound called IPTG. And why do we really need, or why did it show it there? So the trouble is we can't use lactose to efficiently induce our cells. Because remember how I said that the lac operon makes beta-gal, and beta-gal breaks down lactose? So what would end up happening is we add lactose, the lactose would cause a little bit of induction, but eventually it would all get broken down and then no more induction would occur. So we don't want that. So instead we're going to use an analog that looks very, or at least similar enough to lactose and allolactose called IPTG, where the only real difference is the addition of this propene group. So what this means is when we add IPTG, it'll act almost similar to allolactose and bind to the repressor. So it's able to bind to the repressor and knock it off, allowing for the transcription of our T7 RNA polymerase, as well as beta-gal. But the good thing is this beta-gal and our beta-gal cannot break down IPTG. So it's just able to sit in solution and just continuously knock off this repressor to cause the system to constantly make more and more and more T7 RNA polymerase. So now I kind of want to show you a couple of pictures that I took of, I guess, setting up the entire growth cycle. So these I wasn't able to record So I just thought I would take these pictures and leave them here for you. And I'll make sure to upload these PowerPoints so that you guys have these pictures for your reports. But basically, the first step in doing this expression is actually starting with a colony PCR. So that's where we kind of touch one of several colonies and test them using PCRs, very similar to what we did in our, I guess, our first lab or second lab. So the only difference is instead of a gene template, we use the actual colony as the template. And wherever we see a bright band, this is indicative that our gene is in this colony. So once we're sure that we have a colony that has our gene, we take the colony and inoculate it in 1 liter of media and just let it grow until it reaches that optical density of 0.6. Once it grows, we actually induce it with that IPTG for 3 to 4 hours. And here we can kind of see that after 3 hours of induction with IPTG, we can see the major change where we get this huge, huge band, which is most definitely our beta-gal. So this is how we figure out that if we're actually able to produce beta-gal in the first place. So now that we know that our cells have the beta-gal, how do we get them out? So to do that, we're going to utilize cell lysis. So what we basically need to do is open up all of our cells so that we can release all the inner contents. This includes our protein of interest, but also the other proteins that the cell naturally makes to stay alive, as well as their DNA. So cells can be lysed through various methods. The one we'll be doing in the lab, it is called the freeze-thaw method. And I'll talk a little bit more in just a second, but there's also things like sonication, French press, or utilizing a microfluidizer. So in terms of a freeze-thaw lysis, what this kind of just means is we'll be rapidly freezing at a minus 20 degrees Celsius and then thawing at about 4 degrees Celsius. So taking it in the freezer, throwing it in the fridge, and doing this about 2 to 3 times. And the whole main idea is that we're basically, when this ice forms, when we're freezing our cells, the ice crystals are actually able to puncture the membrane and physically open it up so that the contents inside can come out. To kind of aid in this process, we also add an enzyme known as lysozyme. And what this kind of does is it just breaks down the peptidoglycan layer in bacterial cells. So what we'll do is we'll usually take our cells, mix it in, or add some of this this lysozyme, as well as a hypotonic buffer. So remember what this a hypotonic solution, when it gets added with cells, the liquid will want to kind of enter the cell, causing the cell to expand and want to burst. So like a balloon, we're adding this lysozyme to weaken the peptidoglycan, and then eventually when we freeze it, the ice crystals will pop it and everything will just come right out. So you're not going to be able to see it just by me describing it, but once everything leaks out of the cell, the solution becomes very viscous. So to kind of continue with our lysis and actually get our protein, we add a couple key components, and then, so we add like a couple key components, and I kind of want to describe what each one does. So the first one, right after we're done with our freeze thaws and the solution becomes viscous, is we'll add DNase and magnesium chloride. What this will do is chop up the DNA so that the solution becomes less viscous. The reason we want to chop up this DNA is because, remember, DNA is very long, and so it can get tangled, and it ends up getting tangled around itself and all the proteins, and that's what makes the solution viscous. So that's why we need to chop it up. So once it gets chopped up, we also add streptomycin sulfate, which is a salt, and it will precipitate the DNA. Once the DNA is precipitated, we kind of spin it at a very high speed, and this will pull down all the cellular debris as well as our precipitated DNA. So we'll get we're basically getting like a trash pellet. So in our supernatant, we'll have protein. So to that supernatant, we add ammonium sulfate, which will precipitate the protein, and all we have to do is just spin it one more time and we'll get a protein pellet with all the proteins that were made during our, I guess, cell lysis.